Hello, Bacha Party. Hello, friends, dear friends and students. This is Dr. Sunil Kanal, your friend and mentor and guide, taking your 10th standard revision lectures on science along with Anubhavi Foundation. So, let's start our further revisions. Today, we are going to take Science to second chapter, Life Process in Living Organisms, Part 1. The chapter, interesting chapter with 4 marks compulsion or compulsory questions for 4 marks, and with options it is 6 marks chapter. A very nice chapter. So, dear students, let's start the revision now. What is life processes? Now we know that human being. Let's talk of any human being. Let me talk about myself. Okay, so I am living because my different cells, tissues, organs, and organ systems are working in a very coordinated manner. So our organ systems, okay, various organ systems are continuously performing, okay, their functions in a human body or in our cells, along with various systems like digestive, respiratory, circulatory, excretory, and control systems. Different external and internal organs are performing their functions independently and through a complete coordination. Now, if they are working in a complete coordination, what we can see is that we require energy for such different activities, that is the life processes. Now, from where do we get the all this energy? So carbohydrate, fats and lipids are the main sources of this energy, and it is harvested by mitochondria present in each cell. So they will mitochondria, which are the cell organelles, which are the energy or the powerhouse of the cell, they will harvest this energy. They will use, or they will break down these carbohydrates, fats and lipids to give us energy. But as you know, dear friends, okay, suppose you are very hungry and you are alone at home and you want to eat Maggie. ओके, सो यू जनरली सी दैट एट 2 मिनट, ओके, 2 मिनट में मैगी तैयार हो जाएगा, बट मैगी का पैकेट आपके पास है और गैस खत्म हो गया, या गैस पाइपलाइन से गैस नहीं आ रहा है, फ्यूल नहीं है, तो फिर तो आप कैसे बनाओगे? 2 मिनट में मैगी बनेगा ही नहीं, बिकॉज वी रिक्वायर अ फ्यूल टू बर्न दैट nutrients so that we can get energy in the form of ATP, adenosine triphosphate. So our fuel for our foodstuff, for oxidation of our foodstuff or the nutrients to produce energy, is oxygen. Oxygen is necessary for oxidation of the food so that energy can be released. Okay. So all this is coordinated and controlled in our body for life processes. So how much energy we are getting from carbohydrates, which are the chief source of energy for us? So we get 4 kilocalorie of energy per gram of carbohydrates. Okay. Now, living organisms and energy production. Carbohydrates are the food that we consume every day are mainly utilized for production of energy. And this energy is obtained in the form of biological energy, that is ATP, adenosine triphosphate. For that, the chief substrate means a substance which will be oxidized. So glucose is a chief substrate which is oxidized step by step in a cell to produce ATP, that is called as cellular respiration. Cellular respiration occurs among the living organism by two methods: aerobic respiration, where oxygen is involved, and anaerobic respiration, where oxygen is not involved. In aerobic respiration, glucose is oxidized in 3 steps. So, we are going to talk of aerobic respiration, which occurs in human beings, and that is the main thing which we are going to see. So now oxidation of glucose, the first step is called as glycolysis. Glycolysis means breakdown of glucose to form two molecules of pyruvic acid. This is occurring in the cytoplasm of the cells, and it is a common pathway or common process which occurs in both aerobic as well as anaerobic organisms. Now here, if you'll see that it goes in a step by step manner to form pyruvate. Pyruvate means pyruvic acid. Nowadays, we don't write as acids, but we write in the form of their salts, that is pyruvate. So, glucose is broken down in a step by step manner to form pyruvate. Why doesn't it occur in one step itself? It cannot occur in one step because the heat produced will be so much that it may burst the cell. Therefore, there is a step by step oxidation of glucose. Process of glycolysis occurs in cytoplasm. A molecule of glucose is oxidized step by step, and two molecules of each, that is pyruvic acid, then ATP, then NADH2, that is nicotinamide adenine dinucleotide hydrogenase, and water are formed. Molecules of pyruvic acid formed in this process are converted to molecules of acetyl coenzyme A on the membrane of mitochondria, and then they can be utilized in the mitochondria, okay, for the further respiration, which occurs only in aerobic respiration or aerobes. Okay, two molecules of NADH2 and two molecules of carbon dioxide released during this process. Okay, let's start with the next step, that is of the which is only present in the aerobic respiration, that is the tricarboxylic acid cycle or the Krebs cycle or the citric acid cycle. Now, the very important thing is that is glucose was converted to two molecules of pyruvic acid, which gets converted to acetyl coenzyme A on the membrane or in the membrane of mitochondria, and from there it enters the mitochondria for various steps. It goes into a cyclic pathway which is called as a citric acid cycle. And why it is called citric acid cycle? Because the first stable compound form is citrate or citric acid, and then it undergoes various steps in which glucose is the pyruvic acid is completely oxidized with release of energy. It is also called as Krebs cycle because it was Hans Krebs who had described this cycle. And it is also called as tricarboxylic acid cycle because in most of the compounds there is three COH, that is the functional groups. Therefore, it is called as a tricarboxylic acid cycle. Now, in this, the various steps which are occurring, of course, you don't have in details about these steps, but you should know how the things will happen. The first step is a condensation step combining the two carbon acetyl group with four carbon oxaloacetic acid to form a six carbon molecule of citrate, which undergoes various changes, which you can see how the things are happening. It will undergo a lot of changes in which what happens is if you are going for 11th and 12th with bio or biology as a subject, it will be very useful for you. But now you should just remember that the pyruvic acid, after converting to acetyl coenzyme A, enters into the Krebs cycle in the mitochondria and undergoes various steps of oxidation so that ATP is formed. Okay, here, we don't have the details of those steps. You just have to remember that ATP is formed from these various steps. Okay. And that is the most important topic here that you should know about this cycle. Okay. So you can go through the steps, various steps, just for your information, which will help you to understand the cycle which is occurring. And this is a common thing which is occurring in each and every living cell of the aerobic organisms or the aerobes. Okay, so this is what is happening in the glycolysis, and in this way it is called as a cycle because again the products or the byproducts, that is oxaloacetic acid, is regenerated, which will combine with next molecule of acetyl coenzyme A to form citrate. Thus it goes on, and therefore it is called as a citric acid cycle or the Krebs cycle. Okay, now what is the energy currency here? So we have to know that adenosine triphosphate is energy-rich molecule and energy stored in the bonds of by which phosphate groups are attached to each other. So ATP has an adenosine group and it has attached to three phosphate groups, out of which Adenosine diphosphate combines with inorganic phosphate to form adenosine triphosphate, and the energy is stored in that third bond, and this energy is released for our day-to-day activities. These molecules are stored in the cells as per need, and whenever required, we can use that ATP for our day-to-day activities. This is very, very, very important. Okay, so it contains a adenosine triphosphate molecule contains a nitrogenous compound adenine, a pentose sugar ribose, and three phosphate groups. As per the need, energy is derived by breaking the phosphate bond of ATP. Hence, ATP is called as the energy currency of the cell because the requirement of energy for the cell is obtained from there. So now you can see this is an important chart for us, okay, which will help us to understand that better. So aerobic respiration, okay, in first we'll start from carbohydrates, the basic or the chief substrate, that is glucose. It undergoes glycolysis to form pyruvic acid. Pyruvic acid is converted to acetyl coenzyme A, which enters into the Krebs cycle, and then carbon dioxide and water are completely formed by complete oxidation of pyruvic acid, and energy is released in the form of ATP. If we are taking lipids or proteins, now if I take lipids, which are fats, then they get converted to fatty acids, and they are directly converted to acetyl coenzyme A, which enter into the Krebs cycle, and the same procedure occurs. If we are talking of proteins, proteins are broken down to form the simpler amino acids, the simplest form of proteins, which get again converted to acetyl coenzyme A and enter the Krebs cycle, and the steps follow the same way. In the end, we have to understand that aerobic respiration is because of oxidation of lipids or carbohydrates or proteins, where carbohydrates are the chief source of energy, followed by lipids and proteins. If carbohydrates are less in amount in our body, or due to fasting or due to any other reason, carbohydrates are not in resources or their resources have decreased. So aerobic respiration requires oxygen too in order to create ATP. Although carbohydrates, fats, and proteins are consumed as reactants, aerobic respiration is the preferred method of pyruvate breakdown. So this is the preferred method. This is the preferred method. You can see from carbohydrates by glycolysis forms pyruvic acid, and which goes further. Okay, and pyruvate breakdown in glycolysis requires pyruvate to the mitochondria in order to be fully oxidized by the citric acid cycle or the Krebs cycle. The product of this process are carbon dioxide and water, and the energy transferred is used to break bonds in ADP to add a third phosphate group to form ATP, adenosine triphosphate. Okay, that is substrate level phosphorylation, NADH and FADH2. In this way, the process continues. Now let's talk of anaerobic respiration. Anaerobic respiration is the respiration which occurs in absence of oxygen. Here, there are two methods of anaerobic respiration. One is alcoholic fermentation, other is lactic acid fermentation. In both the cases, the substrate, that is glucose, is converted to pyruvic acid by glycolysis, and then it is used up either for alcohol production or for lactic acid production. In alcoholic fermentation, pyruvic acid get converted to alcohol with the help of yeast, okay, or the fungi, and pyruvic acid gets converted to lactic acid by fermentation, especially in absence of oxygen or incomplete oxidation, okay, by erythrocytes and muscle cells whenever oxygen is less, okay. Now, pyruvic acid on fermentation helps in formation of different products like vinegar, vitamins, okay, and various microbes help in the fermentation process of pyruvic acid. Again, this is an important thing which you should know for anaerobic respiration, okay. So different ways of generation of the ATP by anaerobic respiration. We have just seen the process which you can read down. When you go down the group, you will understand that go down through the points we have been talking about this only. Okay, in this way, here the ATP generation is less because it is incomplete oxidation of the glucose or the carbohydrates. Okay, so that is an important point. So now energy from different food components. Okay, so amino acids, proteins, and different amino acids which are obtained, they have different roles. Okay, so amino acids will be used in skin for the production of melanin and keratin. In bones, there is a protein called as osine which helps in strengthening the bones. Then in cells, various proteins of cell membrane and various enzymes are formed by amino acids. Then in pancreas, insulin and trypsin are formed, which are very useful, either a hormone or it is an enzyme here. Then in pituitary gland, various hormones are generated, which are proteinaceous in nature, made up of amino acids. In muscles, actin and myosin, which are the important proteins which will help in the movement of the muscles. Then in blood, hemoglobin and antibodies are all having amino acids. In this way, they are very, very useful. Okay. So we just saw how it is happening. So in proteins also are called as first class proteins because of animal origin, because they cannot be obtained from plants. We get 4 kilocalories of energy per gram of proteins. Okay, but generally proteins are the last source of energy. It is generally carbohydrates and then followed by lipids, which are mainly used for energy. Now, when we are coming to fats, fats are converted to fatty acids and then they are used for incomplete oxidation or oxidation, which will release 9 kilocalories of energy per gram of lipids. Okay, the lipids are generally stored in adipose tissue, which is a type of connective tissue in the body, and also in the fat cells of the liver. Then the fat soluble vitamins A, D, E, and K, okay, are fat soluble vitamins which are requiring lipids for their formation, while vitamin B and C are water soluble vitamins in our body. So we have seen that FADH2, flavin, adenosine diphosphate, and nicotinamide adenine dinucleotide are produced in the process like glycolysis and Krebs cycle. They are used for various purposes. Vitamins like riboflavin, that is vitamin B2, okay, and nicotinamide, that is vitamin B3, respectively, are necessary for the production of FADH2 and NADH2. Very, very important. They will ask this question and they all also ask the odd man out. Clear? You've understood this much? Now, let's talk of cell division. The next topic is cell division. Okay. इधर तक बंद कर पहले. Okay, we have just completed the first half of the chapter in which we are talking of how is the energy requirement for living organisms, especially human beings or aerobic organisms, can be fulfilled and how ATP generation occurs. But the next important thing is cell division. Now, how do cells divide and they help in development and growth of an organism? Because when the fertilization of the occurs of the two egg cells, that is the sperm from the male and the egg cell from the female, or the ovum from the female, a zygote is formed, which is only one cell. It has to undergo divisions continuously to form a new organism, which is a multicellular organism. Similarly, when the reproduction is to occur, the germ cells, that is the reproductive cells, that is sperms and ova, have to be formed. So there are two methods of cell division. The one is vegetative, that is the mitosis, in which the somatic cells are divided, where the reproductive cells are not concerned. It is a division of the body cells or all the organs and tissues of the body. And the second is meiosis, which is a reductional division in which the germinate, or it is also called as germinative division, in which the germ cells are produced or the reproductive cells are produced. So the first is mitosis. Somatic cells and stem cells, that is the stem cells like that of the spinal cord, etc. Nowadays, stem cell therapy is a very important topic. Whenever you can do some Google search, research on that, you can always do it. It's a very interesting topic too. So mitosis is a equational division in which one mother cell or the parent cell divides into two daughter cells, okay, with equal number of chromosomes. Okay, it occurs in somatic cells, that is the body cells and the stem cells. So mitosis is completed mainly in two steps. What are the two main steps of the mitosis? First is a nuclear division, that is karyokinesis, and the other is cytokinesis or the cytoplasmic division. Karyokinesis is completed through four main steps. Okay, that is prophase, metaphase, anaphase, and telophase. The first is prophase. In prophase, what happens is the chromosomes start condensing to form the thick rod-like shaped structures. Okay, so from the network of chromosome network, it will form the rod-like structures or thick rod-like structures, and they appear with the pair of sister chromatids. Okay, you can see that rod-like structures can be seen here. That is, chromosomic pair occurs and they will form sister chromatids. Centrioles duplicate and each centriole moves to opposite poles. Okay, in animals or the human beings also, there are presence of centrioles which will move towards the opposite pole. You can see these centrioles. Okay, they are moving towards the opposite pole. Nuclear membrane and nucleolus start to disappear, and this completes the prophase. Then starts the metaphase. In metaphase, nuclear membrane completely disintegrates and chromosomes complete their condensation and become clearly visible along with their sister chromatids. See, you can see the chromosomes here with sister chromatids attached at the centromere. Okay, all chromosomes are arranged parallel to equatorial plane. Okay, so if this is the equatorial plane, they will arrange themselves parallel to each other, okay, of the cell. Special type of flexible protein fiber, that is spindle fibers, are formed from the centromere of each chromosome and both centrioles. So you can see that the spindle fibers are formed which will attach to the centromere to the centriole, which are very elastic fiber like things. Okay, the next is anaphase. In anaphase, centromere split, and what happens is very important. Thereby, sister chromatids of each chromosome separate and they are pulled apart in opposite direction. So, suppose this was one chromosome with two sister chromatids. One sister chromatid will come this side, and other sister chromatid will move to the other side, and in this way, they will separate out during the cell division with the help of spindle fibers. Separated sister chromatids are called as daughter chromosomes. Chromosomes being pulled appear like bunch of bananas, and they will go to the opposite poles, okay, of the cell. The last but not the least is telophase. The chromosomes which have reached to the opposite poles of the cell now start to decondense, due to which they again become thread-like, thin and invisible. Nuclear membrane is formed around each set of chromosome reached at poles. Thus, two daughter nuclei are formed in a cell. Nucleolus also appears in each daughter nucleus. Spindle fibers completely disappear, and thus the nuclear division gets completed. After the nuclear division is over, the cells will divide by cytokinesis, and the two cells will separate to form two daughter cells. In this way, the one parent cell has given rise to two daughter cells with equal number of chromosome. Okay. In animals, the cell division occurs, okay, with the help of a furrow or a notch, okay, which is deepening inside along the equatorial plane, and they will separate the two cells. While in the plants, a cell plate is formed, and in this way, the two cells can be separated. Now, coming to meiosis. Meiosis is a reductional division which occurs in the uh, plants as sorry yes, in the plants and animals, both in plants and animals, in the reproductive cells itself, or the it is also called as germ cell division or a reductional division. The meiosis can be divided into two main types: meiosis 1 and meiosis 2. One parent cell give rise to four daughter cells after meiosis 1 and 2 is completed, and each will receive half the number of chromosome of the original chromosome present in the parent cell. The meiosis 1 can be divided as prophase 1, prophase 2, prophase 1, zygotin 1, pachytene, diplotin. In that prophase 1 itself, all this is there, dikinesis, and then all this will get over in prophase 1. So prophase 1, you can see there were following steps. If you come to prophase 1, we will not have that much details for it, except for the diagrams you should know. Just remember, prophase 1 is a first stage of meiosis 1, again, which is divided into five phases. Remember the name of the phases: leptotin, zygotin, pachytene, diplotin, and dikinesis, where along with the sister chromatids, there will be a crossing over between the homologous chromosome. Crossing over means what? Some part of the sister chromatids are exchanged between two homologous chromosomes. What are homologous chromosome? Two similar set of chromosome having same type of genes are called as homologous chromosomes. So five stages of prophase 1, that is leptotin, zygotin, pachytene, diplotin, and dikinesis. Very important. Okay, which for your information has been explained here, so that you can just go through it for your knowledge. It is not there in details for you in the portions. Okay, you should only know the topic properly. Okay, so this topic can be understood. You can just pause the video, you can see them, and you can understand them basically for your knowledge. Then comes after prophase 1, metaphase 1. The same things will occur in metaphase 1. The chromosome line up individually along the metaphase plate. It's metaphase 1. Then anaphase, the kinetochore microtubules shorten, which were produced because of the centrioles, and which are having a that is the uh, spindle fibers, but they will pull the whole chromosome along with both the sister chromatids towards the opposite poles. So full chromosome of this along with sister chromatids will separate. So half the number of chromosome will reach here, half the chromosome number will reach here at the telophase stage. Okay, and after that the nucleus is formed and the cell will divide. Okay, that was in the meiosis 1. In meiosis 2, which can be said that after the two daughter cells are formed in meiosis 1, having half the number of chromosome, meiosis 2 is like mitosis. Each daughter cell will now divide to two more daughter cells, okay, with same number of chromosome which they had received in the meiosis 1. So it is like a equational division. In the end of meiosis 2, the total four daughter cells are formed from the one parent cell. Therefore, it is called as a reductional division with half the number of chromosomes. So, friends, this was something about the chapter living organisms, life process in living organisms, part 1. Now we'll be discussing certain questions with you. Okay, and we'll be talking about certain important questions and discussing how to write them, so that you are well versed with your chapter. we'll be discussing the questions now.
Now let's see the certain questions from the textbook as well as the frequently asked questions in the boards on this chapter. As we had discussed, now see the first question is complete the following chart. Now here uh, they have given two or three boxes which are empty and which have to be filled for 2 marks, which is a very simple question. You have to actually rewrite that full thing in which the boxes and the flow chart has to be drawn with pencil, and you can write inside with the pen. In that you can see that glucose is used up in glycolysis to get converted to pyruvic acid. Pyruvic acid on fermentation, the products will be vinegar and vitamins, etc. And pyruvic acid on fermentation also gives me lactic acid, which is on the right hand side, carried out by erythrocytes and muscle cells. And if it is fermentation, it will produce alcohol, which is carried out by yeast. In this way, you can complete the chart and you can get full 2 marks, which is a very, very simple question. Let's see the second give reason now. The second give reason is our muscle cells perform anaerobic type of respiration during exercise. So give reason. This is a second question which we are discussing after that flow chart. Our muscle cells perform anaerobic type of respiration during exercise. Give reason. The reason is very simple. Whenever we are doing exercise, the muscles require more oxygen. But in the absence of sufficient amount of oxygen, cells, they may switch to anaerobic mode of respiration to get enough of ATPs. During exercise, there is a shortage of oxygen for energy production. Therefore, the muscles have to perform anaerobic respiration, for which they will be doing it anaerobically, and they will undergo lactic acid fermentation. So incomplete oxidation of glucose will occur and lactic acid is formed. Thus, the muscle cells show anaerobic respiration.
Thank you for your patience. Thank you for listening. I hope you have understood every bit of this chapter in details, and you will love to read this chapter more and more. Okay. Along with Anubhavi Foundation, it is your friend and mentor, Dr. Sunil Kanal, always at your help. Thank you very much, and God bless you, Bacha Party.